eukaryote gene regulation. There are a number of things to keep in mind when going from the very simple, very elegant model systems that we looked at. We saw the lac operon, we saw the trip operon, but there are a number of physical features in eukaryotes that necessitate changing over how regulation occurs. Eukaryotes have a nucleus. That means no attenuation the way that bacteria are able to do. Eukaryotes have long-lived messenger RNA. That means that in a bacterial cell, when a bacterium wants to stop making an enzyme, all it has to do is shut down transcription. Once you shut down transcription, you stop making the enzyme within a couple of minutes. You make a message for an enzyme, and it lasts hours or days, which means that you have to have a whole elaborate set of mechanisms to regulate message translation expression. And finally, not really finally, you are multicellular. But not all eukaryotes are multicellular. Number one and number two are true in general for eukaryotes, but there are clearly single-cell organisms like yeast, like protozoa, like algae, that are single-cell and behave a little differently. Your problem is that for many of your genes, that gene has to be regulated differently in muscle cells, nerve cells, skin cells, right down the line. And so there are some genes, especially developmental genes, that can have dozens of different regulatory modules just controlling a small little gene. And so we'll see that in the next chapter that we get to, developmental genetics. So all of this means that you regulate gene expression. So we're looking a little broader now. The full gamut of gene expression, you regulate that at the level of transcription, what we're going to cover today, but you also regulate it post-transcriptionally, what I just mentioned, what we have gone through before. Splicing is a mechanism, a post-transcriptional mechanism to control how a single gene can give rise to multiple proteins. And what we just talked about, you have very extensive systems of translation level control for your messenger RNA in your cytoplasm. So that's the big picture. We're going to take one example. We did two in bacteria. We're going to take one example of eukaryote gene regulation as sort of the paradigm for how typical genes are regulated. And that is the yeast GAL genes. So GAL stands not for gals, but for the sugar galactose. It's how yeast regulate the genes that break down the sugar, the complex sugar galactose, in order to be used as a sole carbon source. So the yeast GAL genes are sort of equivalent to that cell the way that the lac genes are used in E. coli, which means that the yeast GAL genes are positive inducible and under glucose control, metabolite repression. So positive inducible is a little bit different than lactose. Lactose was under negative induction, but inducible means the same thing. Inducible means that yeast is not going to turn on these genes to break down the sugar unless the sugar is there. We saw how E. coli does it. Most genes in eukaryotes are regulated by positive regulation. We saw positive regulation in bacteria, but in bacteria, most genes are regulated negatively. The reason we don't have to do that is because in the nucleus, we have chromatin. And as I've said over and over, chromatin changes everything that happens in the nucleus. So we can consider chromatin, closed chromatin, as being the ultimate on-off switch for most genes. Bacteria do not have that. They have to actually go in and shut down gene by gene by gene. So for a lot of reasons, the yeast regulatory system is going to look different. We're going to talk primarily about positive inducible control. I'll just mention briefly that catabolite repression, that is glucose regulation, is totally and completely different in yeast, actually in you as well, than it is in E. coli. Remember, in E. coli, it's cyclic AMP. That's the message for glucose control. You use cyclic AMP for lots of things, including memory, but not for this. And so glucose control in eukaryotes is by phosphorylation. We are not going to go into it any further than that, other than to say that catabolite repression is just totally and completely different. Kinases and phosphatases determine whether a gene system will be off due to catabolite repression. So that's the big picture. Any questions on the details, on the big picture before we look at the details? All right, so we're looking at the yeast gal genes. There are actually a whole bunch of yeast gal genes. We are just going to look at two, gal 1 and gal 10. And so they're really close by. I put the promoter, the start of transcription on there. I'm going to leave the promoter off. I'm going to leave the P off because the promoter in the normal system is buried in a nucleosome. So this is part of chromatin control. Just like in E. coli, yeast does not normally see the sugar galactose in a situation where it has to turn these genes on. So like E. coli, these genes are kept off most of the time. The other important difference in yeast compared to you, if you look at an interphase cell in your body, most of your DNA is in the form of the 100 angstrom and 300 angstrom fiber. Very, very little is in the form of beads on a string. So that's going to become important when we look at developmental genetics and that system. But in yeast, yeast is really unusual. Most of the DNA is beads on a string. And so here we have a short section of beads on a string. The region between these two genes is called a UAS. So a UAS stands for upstream sequence. This is the sort of equivalent to higher eukaryotes enhancers. So the regulatory region for this pair of genes, notice that they are divergently controlled. We have a central regulatory region, and one gene is transcribed to the left, one gene is transcribed to the right. You know the rules. We're talking about the two different strands of the double helix. Any questions so far? Yes?
When you have the upstream activation sequence, what's that after SET?
That's an approximate,
Oh, okay.
that the UAS is approximately equal to enhancers. So you have enhancers. You don't have UAS sequences. There's some small technical differences that we're not going to get into. But these are binding sites for regulatory proteins. Eukaryotes typically do not have operator sequence. Remember, in E. coli, the binding site for the regulatory protein was an operator. You don't have operators. We work through different terminology and there are different mechanisms for action. Any questions so far? Anybody else? Okay, the important sequences in this UAS, we have a change colors. Ooh, this is nice and bright. So these blue boxes are DNA sequences similar to an operator, but not. And so there are 4 regulatory DNA binding sites in this UAS sequence that we are going to focus on. The major proteins involved in regulation is first the protein GAL4. So GAL4 is the positive ultimate regulator. So I'm going to put GAL4 on each one of those sequences. It's always there. GAL4 is a positive regulator. It has what is called an acidic patch. That is a protein domain in the GAL4 protein that has acidic amino acids, and that is responsible for the GAL4 protein bending over and contacting the RNA polymerase at the promoter. Right now the promoter is hidden. There's no RNA polymerase there. The promoter is buried. GAL4 is the major on button here. The second protein is GAL80. Oh, I should change colors. GAL80 is the negative regulator. So this is the system in a normal cell under normal conditions. GAL4, the positive regulator, is bound to the UAS. It's ready to go, but it's covered up by GAL80, so it cannot turn on the 2 promoters. In other words, we have to get rid of GAL80 if we're going to turn on the 2 genes. Any questions yet? Gal 3 is a negative regulator of gal 80, which makes it ultimately positive for gal 4. Okay, so we have a negative of a negative. If gal 80 is a negative regulator of 4, and you inhibit gal 80, that's ultimately a positive regulator of gal 4. Wording is kind of tricky, but that's just biology. Biology is sometimes complex. Does everybody see how this works? All right. So remember, we have cytoplasm and nucleus. And we have nuclear pores. So again, we now have the full system of gal 80 with the regulators. We have one more wrinkle, and that is there's a promoter out here which makes a non-coding RNA. Notice that it's backwards through gal 1 and on the same strand as gal 10. It turns out 2% of your genome is protein coding, but almost 200% of your genome is transcribed. You have non-coding RNA all over, including sense and antisense. So here we have both strands transcribed, one going that way and one going that way. This is pretty much the rule in eukaryotes. Tons of non-coding RNA, most of which haven't been figured out what they're doing. So we have sense and nonsense over the genes, and in between the genes, we have transcription. This non-coding RNA serves as a platform to repress, to keep these histone octamers over the promoter. When we induce, when we turn on the gene, that's going to destroy the non-coding RNA so that we can remove the histone octamers over the promoter and put an RNA polymerase in there. So the whole thing is set to go now. We have gal 3 floating around in the cell. We have gal 80 bound to gal 4. Gal 4 is bound to the UAS. And the whole thing is not working because we have histone octamer over the promoter facilitated by this non-coding RNA. Now we're going to induce. Any questions so far? When we add gal, we're going to change the shape Gal3, which is the negative regulator of Gal80. The thing is, with 80, that I really can't show easily, is that 80, all of it, keeps coming on and off. So Gal80 is not glued down to Gal4. It's a biochemical system. It has a binding constant, so it keeps coming on and off Gal4. Now that we have added galactose, we've added the inducer, we've changed the structure of Gal3, when Gal80 comes off, we're going to trap Gal80 off of Gal4.
So when Gal80 comes off, Gal3 attaches to it, or does Gal3 grab it from on top of Gal4?
The prevailing models seem to suggest that 3 grabs it in the nucleoplasm and cytoplasm. Because it's coming on or off by itself, it doesn't need to be pulled off. Anybody else? So now we no longer have the negative regulator of Gal4, and now you can remove the octamer. I have more colors. And so now we can bind RNA polymerase So what's going to happen DNA is going to bend, physically contact the polymerase. That's how all your genes work. Your enhancers bind proteins. The enhancers can be thousands of base pairs away. The DNA is going to fold up. All of your genes work like this. The DNA is going to fold up like a pretzel. The positive regulator is going to make physical contact, I'm going to be vague deliberately here, with elements at the promoter, including mostly the transcription factors. I'm showing it at the RNA polymerase, but mostly with the transcription factors. I don't want to make this thing too messy. This is how all gene regulation works in eukaryotes.
What causes the nucleosome to come off?
So once you take 80 off, there's going to be a whole bunch of changes. Changes to the chromatin. Once that comes off, I forgot to take this off too, this goes away really fast. And so you don't have the enzymes that hold the nucleosome there. You have other enzymes modifying the histones so they come off the DNA. Yes?
So once now is removed, the octamers come off at the same time.
Once Gal80 is removed, the non-coding RNA goes away, which means that the enzymes which are holding the nucleosome in place, loosely translated, they go away, so the nucleosomes come off, the promoter is freed up, the polymerase, but we bind the transcription factors. Remember, the transcription factors bind first. Your polymerase is illiterate. Then the polymerase binds, and then 4 bends over to make physical contact with all those proteins at the promoter to start transcription.
Is there an RNA polymerase at each end?
Yes, there is. The same thing is happening at both ends. Yeah, I have that up here. Yes.
Does it bend over to make the polymerase go, or does it bend over to make sure nothing else can come back in?
It bends over so that the positive regulators contact the proteins at the promoter, and that activates the polymerase. A very, very different scheme than what we saw in bacteria. Yes?
Will you repeat what you said the positive regulators?
But so the positive regulator, which is Gal4, bends over, so we're going to bend the DNA, bends over to contact the transcription factors and polymerase at the promoter.
You've got multiple Gal4s there.
Yes?
Does each one contact the same promoter or different promoters along the line? Is it consecutive?
That's an interesting question. I have not seen it answered. I've drawn it accurately, but there's a group of 3 and a group and 1. It's not known if 3 go this way and 1 goes that way, or if 2 and 2, it simply isn't known. And it may bounce back and forth because a lot of that occurs in real systems. But that would take, you got to look at a single cell at a time, which is difficult, but people are starting to do it. Any last questions on how this works? Remember, this thing is in chromatin. Any questions which may pop up on the 3rd exam that asks you about this, we're showing chromatin because we have those nucleosomes there. Everything that you do in a eukaryote nucleus has to take into account chromatin. Chromatin runs everything.
So the Gal4 is like sitting on top of the promoter?
No, Gal4 is sitting on its sequence in the UAS. When it bends over, it's still not going to bind the promoter. It's going to bind to the protein found in the promoter. Anybody else? Yes.
Are you trying this to be done in the cytoplasm or in the nucleus?
Why did I do that? I just put the label backwards. But no, I really did mean it's happening in both the nucleus and the cytoplasm. 0 that one. Yes.
When it bends over, does it take with it the UAS sequence as well?
Oh yeah, it's going to be bound to the UAS. And so the DNA is going to end up looking like a pretzel. But that's the absolute rule in eukaryote regulation, is multiple regulatory proteins binding tens to thousands of kb away are bending over to contact promoters and working. Huge regulatory regions compared to bacteria. Absolutely, yes. Yeah, the drawing is absolutely the way I want it. So it's happening out here, and it's happening in there. Anybody else? So we can take the system that we have there and extend it to developmental genetics. Developmental genetics is nothing more than gene regulation over space and time. We are going to look at a few different aspects of developmental genetics. The first one, we're going to ask a very simple question. How do cells become different? So you are a multicellular organism, and unlike a fungus, where every cell is pretty much the same, you are a collection of probably thousands of different cell types. But everybody here started off as an egg and a sperm that united to create a fertilized egg. We all started out from a single cell that then underwent mitotic divisions. We talked about this. Mitosis gives identical nuclei. And so when you started out, you were a 1 cell, and then 2, and 4, and 8, and 16. And at some point, not 16 yet, at some point, you began to have cells that were slightly different from each other. And those differences became magnified. That's the process we're going to start off looking at, is what is the mechanism that creates skin cell and muscle cell, for example? What's the mechanism that makes your head different than your feet, your back different than your tummy? That's what we are going to look at in a very well-developed system in Drosophila, because that's where this was all worked out in well, actually from the 1940s right on through. The breakthrough came in about the 1970s when 3 Drosophila geneticists won the Nobel Prize for figuring out pretty much what we're going to talk about today. And so the question that we are going to ask, very simple question, how does a group of cells with identical nuclei, remember mitosis, how does a group of cells with identical nuclei end up having different cytoplasms, end up having different genes turned on? Okay, that's where we're starting. We're starting with that. So this is actually a pretty good cartoon of Drosophila. Drosophila is an insect. It's a classical genetic research organism. Probably the majority of genes that have phenotypes have mutations, so it's very easy to work in, very inexpensive to work in. So a female, not easy to tell here, lays an egg, and that egg undergoes development, and that egg becomes segmented so that ultimately that egg becomes a fly that lays more eggs. I'm going to tell you the secret of development and differentiation. Remember that the nuclei are identical. Mitosis is a process that creates identical nuclei. So in order to get different cells, the secret is that the cytoplasms are different. So the cytoplasm of a muscle cell is different from the cytoplasm of a nerve cell, and so on and so on and so on. So now we have to think about this conundrum. The nuclei are the same. How do we have different cytoplasms? And that's the major problem that we have to address. The nucleus runs the cytoplasm because the nucleus determines which genes are expressed and which proteins are made. So the nucleus runs the cytoplasm. But the cytoplasm runs the nucleus. Because what is in the cytoplasm determines what genes will be expressed. So it's sort of a circular regulatory system. But that's what we're going to look at. Any questions on my artistic ability here? All right. So everything really starts off with an egg. But that's not true either. The egg is only where you see the beginning. Because what you don't see is that the egg is made by the mother. And it is the way the mother makes the egg that determines the outcome. And so you all know the egg is the largest cell in any organism. Again, think chicken egg. And so to make an egg requires an incredible investment by the female. The female has to put everything into that egg to run development until that baby hatches out and can start feeding on its own. For different organisms, that's vastly different requirements, up to and including chicken eggs, ostrich eggs, which are enormous. So this is where it becomes interesting, because it's how the mother makes the egg that determines what happens after fertilization. The mother, especially in eggs that are laid, so eggs in mammals are a little bit different. Well, I'll come back to that very, very briefly. But in the vast majority of organisms where an egg is laid and development then occurs, the mother has to put everything into that egg for early development. But it's not simply a matter of dumping stuff in. She has to put everything in, including all the messages needed, all the proteins needed for early development, all the tRNAs, and the biggie, all the ribosomes. And so I told you way back in the middle of the course that you do gene amplification. Actually, half of you do gene amplification. You women cannot make a viable egg with the 300 or 400 ribosomal genes that you have. You just don't have enough genes to make a good egg. And so before you start the egg-making process in your ovary, you do gene amplification. Look that up, it's a term we had about the middle of the semester. You amplify your ribosomal genes up to 1,000 or more copies. And then you can make enough ribosomes for your eggs for early development. I'm sure you all know males make virtually no contribution. It's just a haploid nucleus with a tail. Usually a mitochondria in there for energy and power. And so sperm's cheap, eggs are real expensive. So that's the basics. Now, having said that, you women can't just dump stuff into your egg. It isn't as bad for mammals, but for Drosophila it's critical. So it's not just what you put in and how much, but its location. In the system we're going to look at, location is everything. The female Drosophila can get everything in, all the messages, all the protein, all the ribosomes, everything. And if she doesn't put messages, especially, if she doesn't put all the messages the right location, the embryo isn't even going to hit embryo stage. It's going to die really, really early. And so what we're going to look at, we're going to start at the very beginning with an egg. And what I'm going to describe to you is not how the mother makes it, but what it looks like at the end. The key to differentiation is the cytoplasm. I said the cytoplasm rules the nucleus. The way you get different cells is by having cytoplasm in mitosis not equally divided. So mitosis equally divides the genes. Each cell gets identical genes. But if you want differentiation and development, the two daughter cells will get different sets of cytoplasms. That means the mechanisms in cells are established so that the cytoplasm is not homogeneous. The cytoplasm is very, very differentiated. Again, think chicken. You have the yolk, which is in the cytoplasm, segregated differently from the rest of the cell. In Drosophila, we don't have a big yolk like that. The yolk is uniform, but I'm going to show you that regions of the cell have different messages. And that's the key. It's also the key to bacterial differentiation. It's also the key to sporulation, is dividing up the cytoplasm not equally. Not equal cytoplasm is the whole key. You can have everything in there, but if you don't divide it up right, it dies. You had a question, I cut you off?
You answered it. You answered it.
Okay. Anybody else before I start the description? Yes?
You can just summarize like the take home there, like
The
location and all that.
take home is that it's where the mother puts the messenger RNA. Everything else is going to be uniform. It's messenger RNA because early eggs have to distinguish anterior from posterior. They have to distinguish front from back. Different species actually have very different mechanisms. Sometimes it could be as simple as where the sperm enters the egg. Drosophila, it's all predetermined, it's laid down by the mother.
Wait, where the mother puts the mRNA where?
In the egg.
In the egg.
In the egg. That's what we're talking about.
Front versus back?
I'm sorry?
Front versus back?
Yes, in this case, it's front versus back.
In the case of flies?
I'm sorry?
In the case of flies, you say?
In the case of Drosophila.
Because I mean, that's not.
Your question?
Oh, no, I was stretching.
Anybody else before we start the process? So what we're going to describe, we have this egg. We have to distinguish anterior from posterior, dorsal from ventral. We're going to do that with 3 different messenger RNAs. What we're going to do, what the female does, is designate anterior with a message called BCD, which stands for bicoid. So, okay, let me explain one little thing. This work that I told you about in the 1970s was done in Germany, a collaboration between a German investigator and an American investigator. And so many of the names that we're going to do are going to be German names. So just like the signal transduction pathway, where a lot of the names of the genes are in Japanese and transliterations, a lot of the names are going to be German names.
And so the female designates message to where?
To where I'm going to show you. Anybody else? So bicoid message is going to be laid down in the anterior part of the egg. Not going to mechanism, if you're interested in that, I think it's in your book, but you can stop in and we can talk about it. In the back, we have another messenger RNA called NOS, which stands for nanos. The messenger RNA is put in the egg. Messenger RNAs have an open reading frame. Remember, they also have a 5' untranslated region and a 3' untranslated region. Those regions have a lot of information. Among the things in a messenger RNA, besides the open reading frame, are address labels. Real, literal address labels in terms of an RNA sequence coded in the gene that binds proteins that send the message to different parts of the cell. The bigoid message has an address label that says put it in the anterior. Nanos message has a label that says put it in the posterior. And that's how we're going to form the anterior to posterior axis. So we're going to have two axes, AP and BV. Any questions? In the ventral portion, we're going to have a message called dorsal. It may sound strange. We have a messenger RNA, and the message is the same as the gene name. So we have a gene that codes for a message. It's called dorsal, but it's laid down in the ventral part of the cell. What you have to understand, and you should remember this, is that genes are named for the phenotype. What that means is if you misplace the dorsal message, the bottom looks like the top. It looks like ventral tissue. And so all these genes go back to classical times where the gene was named for the phenotype of the embryo or fly. So a dorsal mutant is ventralized because you need the dorsal message to specify dorsal polarity. Is that clear to everybody? Kind of complicated, but it makes sense if you think about how genes are named by the phenotype. So the gene is named for the phenotype. If you knock out the dorsal gene mutation, the whole embryo becomes ventral. So the dorsal message is making a protein which turns that ventral into ventral tissue without the rest. Anybody else?
So, those are the dorsal and nanos, those are names of the...
So that, well, the names of the genes and the message is called the same as the gene. So we're only going to be concerned with 3 different genes. So these genes are expressed during egg development as the female is building the egg. And if she makes a mistake and doesn't pack down the message where it belongs, the embryo isn't going to have any chance to do anything. It's going to die almost right away. And so what we're going to have now we can have fertilization. Haploid nucleus, and so there's half a nucleus there, and half a nucleus there. That's going to be, well, it normally happens as the egg passes down the oviduct. The egg passing down the oviduct is going to be the trigger to start everything off. Remember, these are messenger RNAs. the messenger RNA is going to be translated and what you're going to see now is a concentration gradient. So graphically, we have high concentration of protein near where the message is laid down. The protein is translated from the message and diffuses out. So the further out we go towards the middle, the lower the concentration of protein is. So we are establishing a protein gradient there and here. Is my artistry okay? Everybody see what's happening? Message is laid down, tacked down to the ends, and then the message is translated after fertilization, and the protein diffuses out. Distance. Yes.
The distance from mRNA to this ends.
Oh, just the distance from the anterior tippity-tip end to the posterior tippity-tip end, which is concentration gradient. Kind of. I'm going to short circuit that and just show the result. Dorsal's more complicated. Don't worry about dorsal. Anybody else? Yes.
So it's
Louder.
translated to protein and proteins.
And so the protein is diffusing out. The message can't move if it's tacked down. Protein's small and diffuses out. And so the whole thing is also timing is everything. But the trigger is fertilization, so that's not an issue. Anybody else? Okay, let me put dorsal out here. And so the same thing is happening. Dorsal protein, but the mechanism is different. Don't worry about the mechanism unless you want detailed information to stop in and we can talk about it. But it's different from Nanos and Big Boy. And so we have 3 sets of proteins, and we have defined the anterior, posterior, and dorsal ventral. So the anterior end becomes defined by having a high concentration of bigoid protein. The posterior is defined by having a high concentration of Nanos protein. The dorsal is defined by having a high concentration of dorsal protein. But dorsal is done differently. Now, dorsal and bigoid are gene regulators. So what's going to happen now this is ultimately going to give rise to segments. Because bigoid and dorsal are gene regulators, that means that different genes are going to be turned on in the anterior versus posterior. So you can think of these boxes now as cells. Drosophila has some peculiarities that mammalian cells have, but you don't have to worry too much about that. And so we've got these cells under control there. We have the bottom under control of dorsal, and we have the posterior under control of nanos. And so what we have are unit cells that have different combinations and concentrations of gene regulators. So that's the first key in the developmental process, is different combinations and concentrations in different regions of the cell. And so the thing starts off regionally. So this is all about maternal genes controlling the first step. And if the maternal genes are messed up, everything dies an hour or two after fertilization. The mother's got to get the placement right.
Is the dorsal becoming less concentrated towards the center as well? Because it looks kind of like...
No, it's just I got tired of putting triangles and circles. From the ends and the bottom, it's going to be a pretty uniform gradient. I mean, the gradient shapes are pretty good, actually. So that's about what we Anybody else? Any questions? Yes.
So right now, all these cells are the product of the female's part mRNA.
Well, so remember, yes, all these cells, now these are embryonic cells. All of these cells have absolutely identical nuclei, but this cell has a different cytoplasm from that cell. This cell has, well, different combinations. This cell is different from that cell. So it's hard to get all the details, but the key is combinations and different concentrations of these two gene regulators. Nanos is not a gene regulator, don't worry about that. And so we are now in the position, those are maternal genes. Bicoid, dorsal, and nanos, those are the mother's genes that put those messenger RNAs in. So we are now in the second stage, and that is embryos' genes is now creating segmentation. Remember that fly is a segmented organism. There are head segments that are all squished together. There are thoracic segments, and there are abdominal segments. So we have abdominal and thorax and head segments. Those segments were all differentiated during this process. Different cells in different stripes now have different combinations and concentrations of gene regulators. So we have divided this egg but it's still just an egg. And it do not have the right number of segments there. Do not worry about the number, but they're in the right locations. And so the mother was responsible for the initial distinction between regions. The segmentation genes are now turning on, so different segmentation genes will turn on there and there because we have different combinations of gene regulators in these 2-unit cells, the way I have drawn them. We are now in the middle, the second phase of embryonic differentiation. How your head looks different from your foot. You don't think of yourself as being segmented. You have the exact same set of genes. You use them differently. If you think you're not segmented, run your hand up and down your spine, and you'll feel those segments. You use these genes for different things than the fly does. Any questions on how segmentation is determined? So the maternal genes regulate and turn on the segmentation genes because of different combinations and concentrations of the maternal genes. Now that we have the fly segmented, the next step is to define each segment. By that, so back here we have the segments. Now we have to say, okay, this segment and that segment are going to give rise to head structures. This, and this will give rise to the thorax. The thorax has wings and legs. And so the next step from the segmentation genes, we have the homeotic genes. They identify segments. So again, different combinations and concentrations. Now we have more regulators. We have segmentation gene regulators. If you look in your textbook for those details. The segmentation gene regulators and some of the original initial maternal genes, we have a lot of different gene regulators, and they now identify the segment. And okay, these first few segments are head. That means we turn on different structural genes, eyes, antenna for the fly, mouth. We turn on different thorax genes, wings and legs, and abdominal genes for a lot of the internal structures, ovaries, testes, gut, all of that are turned on differently in different segments. It's all about different combinations and concentrations of gene regulators set up initially by that distribution. Anybody have any questions? So what do you think would happen if we had a mutation that knocks out one of these sets, a segmentation gene mutation that's now going to be homozygous, because these are active in the embryo. These genes are not embryonic, they're maternal. What do you think the visible effect of a segmentation gene mutation would be?
you'd have different things in the wrong place?
Can I hear you?
Things in the wrong place?
No, not things in the wrong place.
Loss of a segment.
Loss of a segment or duplication of a segment. Both of those are pretty common. What about the homeotic genes? Homeotic genes identify the segment. What do you think a homeotic gene mutation would look like? Wings on the head. That's the classical example. That was your answer for the wrong question. So a homeotic gene, the classical homeotic gene is called antennapedia. Probably found back in the 30s. Instead of having nice antenna, a homeotic antennapedia mutant, it will put legs on the head. It will, because we're identifying what a segment will make. If you express the wrong gene, and you express a gene for thorax in the head, you will make thorax parts. And legs are on the thorax. And so the classical homeotic gene mutation is replacing the antenna with legs. The fly has just an extra pair of legs. It usually doesn't hurt the fly very much. They seem to get along just fine. Any questions so far? All right, now, for these homeotic genes, talk about turning genes on and off. But the homeotic genes in particular, once you identify this segment of cells as thorax, you want that segment to be thorax for the rest of the fly's life. And so it's not just turning on leg genes here and turning off antenna genes. What you want to do is epigenetic control. Once you say this is a thorax and you set the regulatory genes, you want those genes to know they're thorax for the rest of the fly's life. That's epigenetic regulation. Where have we seen epigenetic regulation in a big system before? What?
The sex chromosome regulation.
Yeah, exactly what?
Female X chromosome.
That's it. What's it called?
Bar body?
Bar body. So, and it happens early in development. When you were an embryo, you don't even remember, every cell flipped a coin. Instead, am I going to use my mother's X or my father's X? And you shut down one of those. And then you fixed the chromatin epigenetically so that with every cell division, every cell resulting from that one cell that made a decision, every cell has the exact same X chromosome shut down. Well, you've got to do that for lots and lots of places other than the X chromosome. And where the fly does it is in the segments, in the identity of the segments. When the fly is an insect, it's going to go through metamorphosis. And when it goes through metamorphosis and comes out of metamorphosis, the larvae becomes a fly. That fly has to know just what those segments were from when it was a larva. And so the genes, a lot of the genes have to be set epigenetically. Any last questions on this? Okay, so now we're going to skip time. Right now we're in early embryonic development. We're not even up to a fly yet. That's days later. We're early embryonic development. So we're going to skip ahead time-wise and species. And we're going to go from Drosophila to one that you know well called humans. We are going to look at two different cell developmental areas in human development, and that is how you determine the cells that will give rise to your red blood cells, cells that carry the oxygen around. When we've done that, we'll look at how you determine one of your white blood cell types that makes your antibodies. Understand that the early mechanism for your red blood cells and white blood cells are pretty much exactly this, just modified for species and at a later time. So the whole thing is the same. Your maternal genes gave rise, not the segmentation genes, but the genes which identify different regions that the egg is going to become as the egg goes through 2-cell, 4, 8, 16, and so on. Drosophila really has a different kind of development early that we skip over. But it's not important for the mechanism. And so we are going to jump ahead in time and look at an early embryo, already pretty visible, and how that early embryo decides which cells are going to become red blood cells and make the protein hemoglobin. And it's going to take a different set of cells and make a decision how that different set of cells will make the antibodies that you use to ward off viruses and bacteria. Any last questions